Good morning, dear students. Today we are going to continue the DNA replication chapter. In this section, we will start with the replication machinery. What it means, replication machinery? That means the whole set of so many things that may include some enzymes, some proteins, some other things. The whole thing required to complete DNA replication that forms the replication machinery. Then let's see what are the prerequisites one by one. Of course, template DNA should be there. Otherwise, replication is not possible. Template DNA is a must. So that is the first one. Second, for preparing anything, we need ingredients. So here the ingredients or the building blocks are the precursor nucleotides. So as this is DNA, here the building blocks or the ingredients are deoxyribonucleotides. Deoxyribonucleotides. So many different types of deoxyribonucleotides are necessary, like deoxyadenosine triphosphate. I will write deoxyadenosine triphosphate, then deoxyguanosine triphosphate, deoxycytosine triphosphate, deoxythymine triphosphate, like these. So many different deoxyribonucleotides are necessary. Then next one, which is the mediator. So many enzymes are there to facilitate the DNA replication. Which are those enzymes? DNA polymerases, DNA ligases, helicases, primase, topoisomerases, etc. All have their own individual duties in this DNA replication. We will see what all those duties later. Then some proteins also required, very necessary. For what all reason, what all purpose they are required here? For unzipping the two strands, we know that the DNA is a coiled structure. For replication, need to unzip this one. For unzipping, some proteins are necessary. Then to recognize the origin of replication, origin, a small stretch of DNA, that also we will learn later. To recognize the origin of replication also requires some protein. Then to stabilize this single-stranded DNA requires some proteins. Then after replicating a certain portion, again need to recoil. So to reform the helically coiled DNA molecules also requires some proteins. Then examples for such proteins include sliding clamp proteins, clamp loading proteins, single strand binding proteins, etc. etc. Okay. So we are starting the mechanism of DNA replication in prokaryotes. First step, need to activate our building blocks. Building blocks, which are the building blocks? Deoxyribonucleotides. So initiating or starting anything, we need to boost up all the participants. Likewise, here also we need to activate our participants, that is the deoxyribonucleotides. That means they are in a low energy form initially. Let's see. This is the cell. Consider this is the cell, and inside the cell we know nucleus, and inside the nucleus DNA is there. And using this DNA itself need to replicate. Need to create a copy of this one to give to its daughter cells. For that, we know our building blocks. That is, deoxyribonucleotides are there. Look here. Here there is only one phosphate. That means it is in low energy form. So from this low energy form, we need to activate this deoxyribonucleotides. How we can activate this one? We can activate this by taking energy in the form of phosphate from ATP. We know that the ATP is adenosine triphosphate. three phosphates are present here. From these three phosphates, by taking two phosphates to here, we can activate this deoxyribonucleotide to incorporate to the replicating DNA. So by its own, it will not give this phosphate. It need an agent. That enzyme, there is one enzyme. Its name is phosphorylase. Its name is phosphorylase. So with the help of this enzyme, from the ATP, two phosphate groups are adding to here, and by that way that deoxyribonucleotide is activated. Now it is carrying three phosphate group, and after giving two phosphate group to here, the ATP becomes AMP. So this is the activation first step. Okay. Next, we are going to understand about origins of replication. What is this origins of replication? They are certain region, specific region in the DNA where the replication starts. So, as we are now talking about the DNA replication in prokaryote, here we are taking the case of E. coli. In E. coli, the origin of replication is named as ORI-C. This ORI-C is a 245 base pair long DNA portion in its genome. So, at that point, the replication is starting. Then what is replicon? Replicon means it is that portion, that stretch of DNA and which replicate only under the control of a single origin of replication. Clear? So, replicon means is that stretch of DNA which can replicate only under the control, under the regulation of a single origin of replication. Okay, so in the case of prokaryote, as there is only one origin of replication, there will present only one replicon. But this is not the case in the in the case of eukaryotes. There, so many origins of replications are present, and by that reason, they will present so many replicons. Okay. Anyway, now we can take the case of prokaryotes. Let's see how the replication begins. So this is the DNA of E. coli, simply prokaryotes, and we can very clearly see the origin of replication as ORI-C here. And first, how it begins? It begins as a protein start to act. That is DNA protein, DNA a protein. And what this protein is doing here? This protein comes and correctly bind on that origin of replication. As it bind on that origin of replication, that point start melting and begins that strand separation. That strand separation, you can clearly see that here, strand separation. Yes, that strand separation begins there after the binding of that DNA A protein. As that strand separation initiated there, then came another protein. Its name is DNA B protein. It also come and bind near to this DNA A protein and continue that strand separation. You can see that here. And as a result, as the separation of strand continued, that will result in the formation of a replication eye or replication bubble. Okay, this is what formed there. Then in this replication eye or replication bubble, we can see two Y-shaped, here one Y-shaped structure, here also one Y-shaped structure. They are called replication fork. Replication fork. So in the case of prokaryote, during the time of replication, we can see two replication fork since a replication eye is forming here. And also we know that the two strands, its orientation is anti-parallel. So because of that reason, when two daughter strands start to form here, daughter strands 3 dash end will face towards this replication fork, while if we take the case of the other DNA strand, its 5 dash end. Its 5 dash end will face towards this replication fork, because the orientation of two strands always be opposite or anti-parallel. Is it clear? The main steps in DNA replication are initiation, elongation, and termination. So, we are moving to the first main step, that is the initiation process. In the former slide, we have seen that the replication I is formed, and replication fork. What is the replication fork? That also we understood. So, after that, what is going to happen? That we are seeing in initiation process. So replication I, we can see replication fork also present here. After this, there come another enzyme. Its name is DNA helicase. Helicase, and its duty, its function is to unwind and twist the DNA strands, the coiled DNA strands. So that's what it is going to do next. So it move down the DNA and untwist and free the DNA strands. So that we can see. So with the help of DNA helicase, this much portion is unwind now. And as because of this unwinding process, the area in front of this DNA helicase, it undergoes supercoiling because of this untwisting done by DNA helicase. Then what can do to relieve the supercoiling? Can be done with the help of another enzyme called topoisomerase 1 and 2. Why is 1 and 2? Because the orientation in which it relieves the supercoiling, there is a difference. It can relieve positive and negative supercoiling. Because of that difference, there are two topoisomerase 1 and 2. Anyway, leave that. This enzyme come and relieve the supercoiling in the DNA strand. Okay. Then we can see that a appreciable portion of the DNA is untwisted now. So to keep that DNA in that form itself is very necessary. Otherwise, it will again undergo coiling. For that, there will come another stretch of proteins called single-stranded binding proteins. So what they are going to do? They rush to the site and bind that single strand. Here also it will bind, but I am not showing that here. Anyway, you need to understand. The single-stranded binding protein comes and bind all these separated DNA strand to prevent its retwisting. Okay. So that's what happened here. And after this will come another enzyme, that is RNA primase. Then what is the role of this enzyme? For initiating the DNA replication, it requires some foundations. Foundation means that foundation is in the form of a short RNA sequence. That short RNA sequence is generated by this RNA primase enzyme. So we can see that it generates that short stretch of RNA, and its name is RNA primer. Like the foundation to our house, here it laid a foundation for the further development, further growth of that new daughter strand. And also this RNA primer provide a free 3' OH end here, and to this end is adding the first deoxyribonucleotide, and after that sequentially further more and more nucleotides will come and bind and will grow the daughter strand. So that is the RNA primer, and it is synthesized by RNA primase enzyme. Okay. And one more term we need to understand, primosome. What is primosome? Primosome, we are giving that name to that particular aggregation or that complex consists of this primase enzyme. Then we know helicase enzyme and also SSB protein. SSB protein means single strand binding protein that we have already seen in the former slide. So all these three, 1, 2, 3 together, we can call it as a complex called primosome. So that term also we should be familiar. If any such question came for the examination, we need to write. Then in the case of this prokaryote, the replication is bidirectional. Why it is called bi? Bi means two direction, not in the same direction. As we look to this structure, we can see two replication fork, and these two replication forks are moving in opposite direction. Means it's untwisting proceed in opposite direction. So that is why here in prokaryote we are telling that its DNA replication is bidirectional. Why? Because that moment of or from proceeding of the two replication fork occurs in opposite direction. Then which is the enzyme that is doing this DNA replication? That enzyme is DNA polymerase. DNA polymerase enzyme. We will learn more detail about this enzyme later in this section itself. Anyway, now just understand that this enzyme is doing the replication. Replication means addition of each new deoxyribonucleotide to the free 3' end. Okay. Next, I am going to take only this portion separately to describe some basic thing regarding the DNA replication. So, I have shown that portion separately here, and look in this region. We know that in the case of prokaryote, there is only one origin of replication and its position is somewhat here. And in this picture, it will be somewhere here. And from that origin of replication, need to start the DNA replication. And we have learned that DNA replication always proceeds in the direction of 5' to 3', because this DNA polymerase can add fresh deoxyribonucleotide to the free 3' OH end only. Okay, then take the case of this template strand. Its end is 3'. So, this DNA polymerase enzyme, it synthesizes the daughter strand in 5' to 3' direction. There is no problem. The orientation is correct, and continuously itself that daughter strand can be formed with a complementary sequence to that of the template strand. Okay. Then take the case of the other strand. Other strand, its end will be 5'. As this end is 5', it is not possible to synthesize a daughter strand continuously in this direction, that is 5' to 3' with respect to this template strand. Why? Because this end is 5' and this strand's end should be 3'. So what is the solution? In the case of this template strand, as the orientation is perfect, there requires only one RNA primer, and to its end continuously daughter strand can synthesize. But here it is not possible. Then what is the solution? Here that RNA primase enzyme lay down so many RNA primer here with regard to this template strand. I'll show you that. First, it lay down one RNA primer, and after that, this DNA polymerase add deoxyribonucleotide to its 3-OH end. This primer's 3-OH end is this one, and the 5- end will be this one. So, to this 3-OH end, this DNA polymerase enzyme add fresh deoxyribonucleotide and generate a small fragment. And that fragment is called Okazaki fragment. Okazaki fragment. And this name is given by two scientists. They are Reiji Okazaki and Tuneko Okazaki. Okay, in this way, so many Okazaki fragments are forming here with regard to the sequence in this template strand. Okay, then after that, this RNA primer at the end of each fragment is removed. Each fragment is removed, and these small fragments of DNA are joined with the help of another enzyme called DNA ligase, which is the enzyme DNA ligase. As here, this daughter strand is formed or synthesized continuously. This strand is called leading strand, and as this is synthesized in the form of fragments, it is called lagging strand. Lagging strand. And one more point, as here, the DNA, daughter DNA strand synthesis takes place discontinuously. This type of replication is also known as semi-discontinuous replication. Okay, next we are going to see the pattern or method of DNA synthesis or the growth of daughter DNA strand using the template strand. In this picture, you can see the template strand, and this is the primer RNA, and we can see it's a 3-OH end, and this is the incoming deoxyribonucleotide triphosphate. That is the ingredient or the building block for this growing daughter strand. And the primer attacks the innermost phosphate of this nucleotide triphosphate and cleave and remove the 2-phosphate group from this nucleotide triphosphate as pyrophosphate. Pyrophosphate and join that nucleotide phosphate to this growing chain using what type of bond? Phosphodiester bond. Phosphodiester bond. Why this bond is called a phosphodiester bond? Because it links two sugar groups. And which enzyme is doing this addition of nucleotide triphosphate to the growing daughter strand? That is the DNA polymerase that we already have learned. And that DNA polymerase can add only to the 3' end of a growing chain. That is why DNA replication always taking place in 5' to 3' direction only. Next, we are going to see another function of DNA polymerase enzyme. This DNA polymerase enzyme has vast array of functions. And among those functions, one function is proofreading. That means that enzyme need to assure whether the incorporated nucleotide to the growing daughter DNA strand is correct or not. So there are two ways, two methods by which this DNA polymerase assure the correctness of the addition of nucleotide. Let's see how. This is the template strand, and this one is the primer, and to this primer will come the first nucleotide, and just simply representing it like this way. It comes, and the DNA polymerase enzyme add this nucleotide to here, and on adding to here, then this nucleotide form hydrogen bond with the base in the template strand. Then which are the two methods by which the DNA polymerase assure the correctness of the nucleotide incorporation? First method, by assessing the fitness of the incoming nucleotide to the active site of the enzyme. Which enzyme? DNA polymerase enzyme. By that way, as a first step, first hand precaution, it can check whether the nucleotide approach is correct or not. Then what is the next method? Next method is the proofreading. Proofreading method. How it is doing that proofreading? It checks whether the hydrogen bond formed between the newly added nucleotide and the nucleotide in the template strand is correct or not. We know that between the adenine and thymine, there should be 2 hydrogen bond. Between guanine and cytosine, there should be 3 hydrogen bond. So, by checking all those things, that DNA polymerase can understand whether the incorporation, whether the added nucleotide is correct or not. If it is not correct, then this enzyme can do another function. That function's name is 3' to 5' exonuclease activity. 3' to 5' exonuclease activity. Why we are specifying this activity? Because DNA polymerase is always adding nucleotide in 5' to 3' direction. Against to that, here it done the proofreading in 3' to 5' direction by doing an exonuclease activity. That means it can cleave the wrongly incorporated nucleotide in the last step of addition. Only the last step. It can't cleave any other nucleotide from the innermost region. It can remove only the last nucleotide added to the growing chain. So by that way, it can do proofreading. Then in that way, it completes that DNA, daughter DNA strand synthesis. And after completing the DNA synthesis, so these are all part of elongation step. After completing the synthesis, then need to remove the RNA primer, because RNA should not be the part of this DNA. So that RNA primer is removed by the RNase activity of, again, our same enzyme. Which enzyme? DNA polymerase. So DNA polymerase have another activity, RNase activity. By that activity, it removes the primer RNA. And the gap generated because of the removal of RNA primer is filled by the DNA polymerase itself by further addition of nucleotides. So even after resynthesize this portion, then also remains some nick, some space here, and this space is sealed by another enzyme, that is ligase, DNA ligase. Okay. In that way, replication is finished, and then the final step is the termination. How that termination is taking place. So let's see that. Termination. In this picture, we can see that entire prokaryotic genome. Here we can see the origin, or EC. And from this origin, we have learned that a replication fork will move in opposite direction. And this one is clockwise direction. And this one is anti-clockwise direction. In that way, it moves, and that both directions will meet here, and in this portion some certain base sequences are there. They are called ter sequence, ter sequence or termination sequence. On reaching this sequence, the newly synthesized daughter strand get unlinked from the parent strand and will form the new daughter DNAs. And for making the things more clear, I will explain you about this ter sequence and how it terminate or stop the DNA replication. So in this picture, you can see two type of ter. So why two types of ter sequence are here? Because we have seen that the fork's movement takes place in opposite direction in each side, and this is clockwise and this is anti-clockwise direction. And here we can see some ter sequence like ter J, ter G, ter F, ter B, and here ter A, D, E, then ter I, H. And look, at this ter sequence, some proteins are present, and this protein has two faces. The side that is facing this side, if it face the helicase enzyme, that means it will not restrict the forward movement of helicase. That means if the helicase approaches this ter in this phase, in this side, this helicase can proceed through this ter sequence. Okay. And if the helicase faces this ter or this protein in this direction, in its this phase, this side, then that means the helicase cannot proceed through this ter sequence here. So that is the importance of two phases. Then look here. Here, as the replication fork move, that means the helicase is untwisting the DNA and it is passing through all these ter sequence because it is facing this side. So it cannot stop the helicase. So the replication will proceed. And on reaching here, the other side will face the helicase that is coming through this direction and can stop the replication, the anti-clockwise replication by here. And then take the case of the replication fork on the other side. That helicase can move easily because it is facing this side only, this side of that protein. Then it moves through this way, and on reaching here, the helicase that is proceeding along this side gets stopped on reaching here. So in that way, termination of the replication in the case of prokaryotes takes place. Okay, I think that is clear to you. And in the next class, we will learn more detail about the nature of DNA polymerase enzyme, because we need to understand more about that enzyme. If I include that portion also along with the same video, the video will become too much lengthy. So it's not interesting. We can't concentrate for such a lengthy video. So that's why I am going to wind up here. In this class we have learned, we got an clear cut idea about the method of DNA replication in prokaryotes. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.